GSE 173789 is an RNA-seq dataset of B cells collected from 14 healthy controls and 23 Siemens patients. You may think this is a simple two-group comparison designed to identify differentially expressed genes between the groups. But if you look at the line graph, you will notice that some samples look different. You can switch the visualization from log-2 ratios against the average expression level to the raw data, in this case, the CPM. You see, those samples lack very low CPMs. CPM, counts per million reads, is a normalized measure of gene counts. If the total number of reads is insufficient, CPM is elevated due to normalization. The distributions of CPMs shift to the right in the histograms of those samples, and the sample numbers are close together, except for one control sample. It might indicate the batch effect. In the PCA results, principal component 1, which explains 24% of variance, is driven by the low total reads, you may assume. So I marked them as doubt. The thoughtlessly identified, upregulated genes are, ironically, at least in part likely be the result of normalization. Even if you remove the doubt samples, there are still samples under the effect. These samples are under the same tree node as the doubt samples. These boundary samples are close to others, but still share the expression profile with the doubt samples. So remove them. Let's look at the tree clustering of samples without the expanded doubt samples. There is a tight cluster of half of the healthy control samples. You might assume there are true healthy controls and already MS-like controls. Or, assume this is another batch effect because their sample number are sequential. You can't determine whether this is a batch effect without tracking experimental notes. Anyway, you must know that the identified up or down regulated genes in MS are the products of this cluster. If you suspect a batch effect, you can remove the tightly clustered healthy control samples. Without them, there still seems to be a weak difference between the control and diseased groups. You can identify the genes that are subtly differently expressed. You don't see any batch effect this time. Now, let's compare the three up-regulated gene lists from the conflicting assumption of batch effects. There are only a few genes in the intersection, as you might expect. The enriched Go terms of the thoughtless version of up-regulated genes include terms related to immune response, which might mask poor analytical processes. The second up-regulated genes, under the assumption that there are true and already MS-like healthy controls, give you terms like B cell activation. The last set of subtly up-regulated genes with no batch effect is T cell activation, along with organelles like the lysosome and the Golgi. In conclusion, data analysis without visual aids can easily lead to inappropriate results. The batch effect in omics datasets is often observed, yet many researchers ignore it.